Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing Delhi University MSc Entrance Examination question paper for the subject Biochemistry conducted in the year 2021. So coming to the first, the scientist who identified the transformation agent in Griffith's famous experiment as DNA was. So it was Oswald Avery by as Avery MacLeod McCarthy experiment. Option 2. The scientist who created the first recombinant DNA molecule. So it was Paul Berg, and he did it by inserting DNA from a bacterium into the virus SV40 DNA in 1972. So the answer here is option 2. Then which of the following pair of amino acids are both glucogenic and ketogenic in nature? So they are isoleucine and phenylalanine. This glucogenic ketogenic amino acids have been asked for other MSc entrance exams also conducted by other universities. You can refer to my channel. I have uploaded so many entrance examination question papers. Then next one. Match the coenzyme in list 1 with their transient carriers of specific atoms or functional groups in list 2. So coenzyme A is acyl group, flavin adenine dinucleotide is hydrogen atoms, pyridoxal phosphate is amino groups, thymine pyrophosphate is aldehyde groups. So answer here is option 1, that is A4B3C2D1. Then glycogen phosphorylase exists in two forms in skeletal muscle. The active form of phosphorylase A is generated from phosphorylase B by. So glycogen phosphorylase, it occurs in two forms, and the more active form is phosphorylase A and the less active form is phosphorylase B. And phosphorylase A has two subunits, each with specific serine residue, and that is phosphorylated at the hydroxyl group of this serine residue by phosphorylase kinase enzyme, and ATP is involved in the reaction, and it is ATP is converted to ADP, and you will get active phosphorylase A from phosphorylase B. So the answer here is ATP dependent phosphorylation of a specific serine residue on each subunit. Answer is option A. Then what are bile salts? So bile salts, they are amphipathic compound that act as biological detergents, converting dietary fats into mixed micelles of bile salts and triacylglycerol. Then bile salts are made from bile acids that are conjugated with glycine or taurine. They are produced in the liver directly from cholesterol. So the answer here is option 2, that is amphipathic cholesterol analogues with detergent properties. Then which of the following diseases are related to coronavirus? So it is MERS. Answer is option 1. Then ball and stick in Rasmol are solid spheres and rods representing atoms and bonds. Yes, that first one option itself is correct. Then given below are events in the cell cycle. You have to, which one of the following reflects the correct sequence of events in the mammalian cell cycle? So the first one to come will be phosphorylation of retinoblastoma protein B. Then comes phosphorylation of lamin A, B and C. Then it will be polyubiquitination of securin, and then association of inner nuclear membrane protein and nuclear pore complex protein with chromosomes. So the answer will be BACD. It is option 4. Then next one. The acid hydrolases of lysozyme do not digest the organelle itself. Why? Because lysosomal membrane is heavily glycosylated, and these sugar molecules, they act like a shield, and they will keep the hydrolyse enzyme away from these proteins. That's it. That is acid phosphate hydrolases. So the answer is option 3. Lysosomal membrane is heavily glycosylated. Which chemical you will use for quantifying DNA? So for quantifying DNA, it is diphenylamine, DPA. Answer is option 2. Which of the following bonds inside a cell can be formed by the COH group of a cellular amino acid? So as the most important, okay, ester bond can be formed, that is COH with alcohol. It will give rise to ester bonds. Then another is the amide bonds, because the asparagine and glutamine, they are amide amino acid and they are formed from glutamic acid and aspartic acid. And in this process, the hydroxyl part of the acid is replaced by an NH2 radical, and the reaction take place in the presence of enzyme glutamine synthetase or asparagine synthetase. So it can form both ester and amide bonds. Answer is option 4. Then next one. A protein sample, when subjected to reducing SDS page, displayed two bands of molecular weight 20 and 25 kDa. So reducing SDS page means they have added bromophenol or DTT, dithiotritol, to the SDS page, to the protein sample, and then they have loaded the SDS page and you got two bands, that is 20 and 25 kDa. So that means there is some disulfide bond present between these two proteins, which is broken by the bromophenol or DTT chemical. Then when the same protein sample was subjected to non-reduced isoelectric focusing, a single band was observed. So it is non-reducing isoelectric focusing, so naturally a single band will be observed. The disulfide bonds were not broken. These observations are best explained by the following relevant reason. The sample either contained two proteins of different molecular weight and same net charge, or one protein with two subunits that differ in molecular weight but share same net charge. That can be the answer, because we don't know whether the sample contains two proteins or it is a single protein with two different subunits. For knowing that, you have to carry out further experimental procedures. So the sample contains either two proteins of different molecular weight. Yes, chances are there that they are two proteins with different molecular weights, or one protein with two subunits that differ in molecular weight, or it can be a single protein of say 20 plus 25, that is 45 kDa protein, which got separated into two heterodimers, or else I can say two different subunits, because these two subunits are linked by disulfide bonds. Is in reducing SDS page, the disulfide bonds got broken and the two subunits got separated into 20 kDa and 25 kDa subunits, protein subunits. Okay. So it can be that, and they share the same net charge. Yes, that is why you got a single band. If they shared different net charge, then you would have got two different bands. With isoelectric focusing. Okay. So first option is the right answer. Then uncouplers like DNP or thermogenin uncouple electron transport and oxidative phosphorylation by. So they are uncouplers, so naturally they will be dissipating the proton gradient. Answer is option. Then next one, an enzyme E has kcat value of 600. Kcat is nothing but your turnover number. Calculate the Km value of the reaction. Okay, when reaction proceeds at a rate of 9.6 micromolar per second and substrate concentration is 40 micromolar. Total concentration of enzyme is 0.02 micromolar. So they have given V0, that is 9.6 micromolar, kcat value is given, substrate concentration is given, and total enzyme concentration is given. It is a direct calculation question, direct formula substitution. So kcat is equal to Vmax by total enzyme concentration. Vmax is equal to kcat into total enzyme concentration. You will get 12 micromole per second. Now we know the Michaelis-Menten equation. V0 is equal to Vmax concentration of S by Km plus concentration of S. Substitute the values there and you will get Km as 10 micromolar. Answer is option 3. Then which one of the following is not a heme protein? So delta aminolevulinic acid synthetase is not a heme protein. It is an enzyme that catalyzes the synthesis of delta aminolevulinic acid, the first common precursor in the biosynthesis of all tetrapyrols such as hemes, cobalamines and chlorophylls. It is not a heme protein, but it is a it catalyzes the synthesis of delta aminolevulinic acid, and the delta aminolevulinic acid, it is a precursor in the biosynthesis of tetrapyrols such as heme, cobalamines and chlorophylls. Then multi-enzyme systems include, so multi-enzyme systems. Fatty acyl synthetases, of course it is a multi-enzyme system. Hexokinase phosphatase lysosomes, no. Answer is option 1. Then the pi of a protein is 6. When the electric field is applied at pH 8, the protein, okay, where it will move. So pH is greater than pi because 8 is greater than 6. So naturally it will have a negative charge, and negative protein, they will move towards the anode. So the answer is option 1. Then next one. In glycolysis, pyruvate dehydrogenase is subjected to feedback inhibition by its products. The chemical compound which might be involved in the process are listed below. NADH, FAD, acetyl-CoA, acetaldehyde. Which one of the following combination of the above chemical compound is involved in the feedback inhibition of pyruvate dehydrogenase? So this is pyruvate dehydrogenase action. It requires COASH, that is acetyl-CoA, NAD plus, and these are the coenzymes that are involved, that is TPP, lipoate and FAD are the coenzymes that are involved. NAD plus is converted to NADH and carbon dioxide plus acetyl-CoA. So the answer is option 4, that is A and C. They have asked which one of the following combination of the above chemical compound is involved in the feedback inhibition of pyruvate dehydrogenase. So naturally NADH will be involved and the other will be acetyl-CoA. So answer is A and C. Then the approximate molecular weight of a product after the translation of a 390 bases mRNA will be. So 390 base mRNA, one amino acid is coded by 3 nucleotides. So 390 will be coded by 130 amino acids. Average weight of one amino acid is 110 Dalton and weight of translated product will be 130 into 110, that is 14,300 or approximately 14 kDa. So the answer will be option 4. Then natural proteins are composed of 20 alpha amino acids. Which of the following statement is true for any of these amino acids in a solution of pH 1.5? Okay, now amino acids, they have got carboxylic and amino group. There are also carboxyl and amino group at the side chains of certain amino acids. The carboxyl groups have a pKa of approximately 2, which is almost equal to the a pH of 1.5. What is being given in the question. So they will not be ionized. That is the carboxylic groups, they will not be ionized. The amino groups they have, that is the side chain as well as the terminal amino groups, they will have a pK of approximately 10 and this is greater than the pH and they will be ionized. So the only group that is ionized will be the amino group. So the answer is option 1, only amino group is ionized. Then next one, the arrangement of nucleotides in DNA can be seen by, they can be seen by X-ray crystallography. 2. Amino acid residues which are predominantly involved in the protein DNA interactions are they will be positively charged because the proteins will be positively charged because DNA is negatively charged. So the answer here is option 4. The amino acids will be positively charged. Then select the amino acid which exists as a diastereomer. So diastereomers, they are not mirror images of each other. They are present in those molecules that have more than one chiral carbon atom. So out of these options given below, isoleucine is having more than one chiral carbon atom. Glycine doesn't have a chiral carbon atom. So you can see that here I have written it as 1 and 2, which are the chiral carbon atoms. Then another amino acid that exists as diastereoisomer is threonine. It is also having 2 carbon atoms which are chiral. So the answer here is option 4, that is isoleucine. Then all of the below given amino acids are optically active except. So optically active amino acid, they contain 4 different groups on the alpha carbon atom. Here glycine, it has 2 groups which are similar or same on attached to the alpha carbon atom, that is H and H. So it is the only one which is optically inactive. Answer is option 1. Then next one. Select the statement, true statement about phosphodiester linkage. So option 2 is the answer. 5-phosphate group of one nucleotide unit is joined to the 3' hydroxyl group of the next nucleotide. Then next one. Lecithins are composed of. So they are composed of glycerol, fatty acid, phosphoric acid and choline. Answer is option 3. Molecules that pass through a cell membrane are most easily are. So they are small and hydrophobic. Cell membrane lipid bilayer is present. It will be the hydrophobic region. So smaller the molecule and more hydrophobic it will pass through the cell membrane. Then which one of the following molecules can be utilized for establishing early evolutionary processes? So it is RNA. rRNA. Answer is option 1. The enzyme responsible for conversion of negatively supercoiled DNA into a relaxed circular DNA. So the enzyme, they have asked for the enzyme that will convert a negatively supercoiled DNA into a relaxed circular DNA. So it is topoisomerase 1. And topoisomerase 2, it introduces negative supercoiling. Then which of the following is not served by the plasma protein? Blood clotting, oxygen transport, hormone binding and transport, buffering capacity of blood. So they have asked which is not served by the plasma protein is oxygen transport. Answer is option 2. Only one X chromosome in the aneuploid female is characteristic of individual. That is, one X chromosome means X0 will be present. So it is usually for the Turner syndrome. Answer is option 4. Then which of the following signal molecules does not interact with cell surface receptors? Insulin, gastrin, glucagon, testosterone. Answer is option 4, that is testosterone. because steroid hormones, they pass through the plasma membrane of the target cell and adhere to intracellular receptors residing in the cytoplasm or on the nucleus. Steroid hormones, they are hydrophobic, so they can easily pass through the plasma membrane, and testosterone is a steroid hormone. Okay, coming to the next question. Coenzymes involved in the formation of acetyl-CoA from pyruvate. I have already discussed this in just a few questions back. You can refer to that. It is thymine pyrophosphate, lipoic acid and FAD. Answer is option 1. Then next one. Match the techniques in list 1 with the appropriate applications given in list 2. Salting out. Salting out is for protein precipitation. Ultracentrifugation depends on sedimentation coefficient. Then dialysis is for removal of low molecular weight impurities, and isoelectric focusing is for pI determination. So answer is option 1, that is A2, B3, C4 and D1. Then next one. There are various subclasses of antibodies found in body fluids and body secretions. Many different functions may be attributed to these subclasses. Given below in list 1 is major function of different subclasses, and list 2 consists of the name of the subclass. It is just like match the following. Choose the correct answer from the options given below. So binds to macrophages by Fc. So it is IgG. A4. Binds to mast cell by basophil. B3. IgE. First B cell receptor, that is IgM. No major specific function other than antigen binding. IgD. Protector of mucous membrane. IgA. So answer is option 4, that is A4, B3, C5, D2, E1. Then next one. 5' end capping of mRNA transcripts in eukaryote involves the following statements. Choose the correct answer from the options given below. Okay, you have to arrange it in the correct order. So first comes removal of gamma phosphate of the triphosphate on the first base at the 5' end. Then comes addition of GAMP to the 5' end. Then 5' linkage between GAMP and the first base at the 5' end. Then addition of methyl group to the N7 position of guanine. So answer here is BACD. Answer is option 3. Then arrange the following events of meiosis in correct sequence. So first is synapses, that is option 1. Then crossing over, then terminalization of chiasmata, and then disappearance of the nucleolus. So here also BACD, option 2. Then identify the correct name of the protein present only in biological membrane. It is influenza hemagglutinin. Answer is option 1. The protein involved in photosynthesis, RUBPase. Answer is option 3. Sialic acid is part of ganglioside. The gangliosides are sialic acid containing glycosphingolipids. So answer is option 2. Then photon capturing moiety of chlorophyll molecule is known as, it is Mg porphyrin. Answer is option 2. Receptors present on macrophages can bind to. So they can bind to Fc region. Answer is option 1. Humoral immune response in higher mammals is involved with. So humoral response in which antibodies produced by the B cell cause destruction of extracellular microorganism and prevent the spread of infection. Now the activation of B cell and their differentiation into antibody secreting plasma cells is triggered by antigen, and it usually requires the help of helper Th cells. So answer here is both B and T cells. Then which one below can break a peptide bond enzymatically? So it is papain. Answer is option 1. During UV irradiation of DNA, the following compound is formed. It is thymine dimer. Answer is option 1. Protein is degraded inside living cell through a key process of ubiquitination. Answer is option 2. The observation that plasma membrane proteins mix after cell fusion provides evidence for the fluid mosaic model of the plasma membrane. The same question was asked for MSc entrance examination question paper of some other university. I don't remember the university, but they have asked the same question. Then several classes of hydrolases are localized in. Hydrolases, they are usually localized in the lysosomes. Answer is option 3. Which cofactor is crucial in catalyzing enzymatic conversion of pyruvate to lactate? So lactate plus NAD plus give pyruvate plus NADH plus H plus. So lactate to pyruvate conversion require NAD plus, and pyruvate to lactate conversion it requires NADH. So answer is option 2. In sickle cell hemoglobin, which one of the following is true? A glutamate is substituted by a valine. That's correct. Option 1 itself is correct. Then cellular membranes are self-sealing in nature. If they are punctured or disrupted mechanically, they quickly and automatically reseal. What properties of such sealing are responsible for this feature? So cellular membranes, they are self-sealing. If they are punctured or disrupted mechanically, they quickly and automatically reseal, join together. So what property of such sealing is responsible for this particular correction of the cellular membrane? So it is due to hydrophobic effect of membrane lipids. Answer is option 1. The next one. The chemical nature of covalent linkage in sucrose is known as. So it is known as O-glycosidic bond. This is the sucrose monomers, that is alpha D glucose and beta D fructose. They join together with the release of one molecule of water through alpha 1 beta 2 glycosidic linkages. This is alpha 1 and beta 2 glycosidic linkage. Alpha 1 of glucose and beta 2 of fructose, and this is the O-linked glycosidic bond or O-glycosidic bond in sucrose. Then match the name of the scientist in list 1 with their achievements in list 2 and choose the correct answer given below. So Watson and Crick, they are for double helix of the DNA. R.W. Holly is for clover leaf model of tRNA. Nierenberg is for deciphering the genetic code. Jacob and Monard for lac operon concept, and Alex Jeffrey is for DNA fingerprinting. So the answer here is option 2, that is A3, B4, C2, D5, E1. Then next one. The blood volume decreased when a mammal was bled rapidly. However, the cardiovascular changes resulting from the hemorrhage could be maintained by the following compensatory mechanism. So the blood volume, when the animal was bled rapidly, the blood volume decreased, and once the blood volume decreases, it leads to reduced cardiac output and arterial pressure. And the body, it has got a number of compensatory mechanisms that gets activated in an attempt to restore the arterial pressure and the blood volume back to normal. And these mechanisms include baroreceptor reflexes, chemoreceptor reflexes, circulating vasoconstrictors, renal absorption of sodium and water, activation of thirst mechanism, and reabsorption of tissue fluids. So, increased cerebral flow, that is not there. Reduction of baroreceptor activity and stimulation of chemoreceptors, yes. Reabsorption of tissue fluid in blood, yes. Increased release of enkephalins and beta endorphins, no. So option only B and C are correct. Option 2 is correct. Then from the given intermediates in fat metabolism, the correct intermediate series is, so fatty acyl-CoA, trans-enoyl-CoA, hydroxyl acyl-CoA, keto-acyl-CoA. Answer is option 1. Given below are two statements. One is labeled assertion and the other is labeled as reason. Bile juice help in digestion of food in small intestine. Bile juice contains sodium salt. So both A and R, that is assertion and the reason, are true, and R is the correct explanation of A. So answer is option 1. Then next one. All the membrane proteins known as G proteins have a subunit that activates adenylate cyclase, hydrolyzes GTP. Yes, that is more correct. Because all the G proteins, they hydrolyze GTP. That is correct. Then next one. Which of the following technique is used to obtain the three-dimensional structure of large proteins at atomic resolution? So it is X-ray crystallography. Answer is option 1. Then rifampicin is an inhibitor of. So rifampicin is an inhibitor of transcription. Answer is option 4. HIV infected patients exhibit preferential decrease in. So HIV, it binds to CD4 molecule on the surface of T helper cell, and then it will replicate within them. This result in the destruction of CD4 plus T cells and lead to a steady decline in the population of T cells. So the answer here is option 1, that is CD4 plus cell count. Answer is option 1. In biochemical reaction, the thermodynamic parameter that reaches a minimum is. It is reaches a minimum, that is free energy, because it should be negatively charged. So answer will be entropy and all will be positively charged, because entropy, maximum disorderliness. So answer will be free energy. It will be negatively charged. So it will be the one which reaches a minimum value. Then which one of the following radioisotopes does not emit beta rays? So it is iodine 125. It emits gamma rays, not beta rays. Then molarity of a solution is moles of the solute per liter of the solution. Answer is option 2. Which of the following is a non-membranous structure? So it is microbodies. Sorry, it is inclusion bodies. Okay, inclusion bodies are non-membranous structures. Microbodies, glyoxysomes, Golgi all have single membranes. Then next one. Ribulose 5-phosphate epimerase is involved in. So ribulose 5-phosphate epimerase, it is involved in the pentose phosphate pathway. Answer is option 3. Proteolytic enzymes synthesized as a large inactive precursor are known as zymogens. Answer is option 2. B cell acquire immune competence in. So it acquires immune competence in the bone marrow. Answer is option 2. Absorbance of DNA solution can be measured using a spectrophotometer. Answer is option 2. The enzyme that hydrolyzes starch to maltose is beta amylase. Answer is option 3. Then prolactin is a peptide. Option 1. Insulin is secreted by the pancreas. Option 4. All these are just direct questions. RNA dependent DNA polymerase. So the DNA polymerase here is dependent on the RNA. So from RNA it is converting it to DNA. So naturally it is reverse transcriptase. Answer is option 4. Statins is a popular class of drug known to inhibit HMG-CoA reductase. Answer is option 3. Which of the following scientist was responsible for the first database in bioinformatics? It is Margaret Dayoff. Answer is option 2. The citric acid cycle in respiration yield. So it yields 1 GTP, 3 NADH, 1 FADH2 and 2 carbon dioxide. Answer is option 1. Which of the following property is not exhibited by an allosteric enzyme? Sigmoidal substrate binding curve. Yes. Adherence to Henry-Michaelis-Menten equation. No. Allosteric enzymes do not show Michaelis-Menten equation properties. Cooperative substrate binding. Yes. Presence of separate substrate binding and regulatory sites. Yes. Then specific functional group in an amino acid undergoes specific chemical reaction producing a typical color that helps in the detection of the relevant amino acid. Match the amino acid in list 1 to their specific test in list 2. So Hopkins call is for tryptophan, A2, polys, histidine, nitroprusside, cysteine, millions, tyrosine. So answer is A2, B3, C4, D1. Answer is option 1. Then next one. In the enzyme catalyzed biosynthetic pathway shown below, what will be the effect of substrate A, B, C and D if enzyme E2 is inactivated? So A is converted to B in the presence of the enzyme E1. B is converted to C in the presence of the enzyme E2. And C is converted to D in the presence of enzyme E3. So what they have asked is, what will be the effect on the substrates A, B, C, D if the enzyme E2 is inactivated? So here E2 is getting inactivated, so C will not be produced from B. When C is not produced, D also will not be produced from C. So B will be produced, but not C and D. Answer is option 3. Next question. A certain molecule inhibits an enzyme by binding only to the enzyme substrate complex, or in other words, binding only after the substrate binds the enzyme. This particular inhibition is typical of which class of inhibition? So it is uncompetitive inhibition, where the inhibitor binds to an enzyme binds only to the enzyme substrate complex, or binding only after the substrate binds to the enzyme. So it is uncompetitive inhibition. Select the true statement regarding nitric oxide. So it activates guanylate cyclase. Answer is option 3. Then which of the following protein is not synthesized as zymogen or proprotein? So pepsin as pepsinogen, lysozyme. Lysozyme is neither synthesized as zymogen or a proprotein, collagen as procollagen, and trypsin as trypsinogen. Then heme containing proteins that function as electron carriers in oxidative phosphorylation and photosynthesis are. So they are cytochromes. They are heme containing proteins. Answer is option 4. A drug which prevents uric acid synthesis by inhibiting the enzyme xanthine oxidase. It is allopurinol. Answer is option 2. Then which of the following statement about enzyme is true? Enzyme increases the equilibrium constant of a reaction that catalyzes. Enzyme do not alter any equilibrium constant. Enzyme do not alter the overall change in free energy for a reaction. That is correct. Option 2 is correct. Then a Ramachandran plot. A Ramachandran plot represents the sterically allowed conformation of a polypeptide backbone. That is correct. Answer is option 1. Then which of the following statement is true? Ribonuclease can be treated with urea to produce a random coil. It is not a random coil, it is randomly coiled. That is more correct. Not random coil. So option 1 is not correct. Then, if one oxidizes random coil RNAs in urea, it quickly regains its enzymatic activity. No. But if one removes the urea and oxidizes RNAs slowly, it will renature and regain its enzymic activity. That is correct. Because ribonuclease is completely denatured upon exposure to urea solution, and in the presence of a reducing agent, denatured ribonuclease spontaneously will refold back to its correct tertiary structure with the full restoration of its catalytic activity. And the refolding of ribonuclease is so accurate that it will resemble a native ribonuclease itself. So the answer here is option 3. Then next one. Arrange the following steps of protein synthesis in order in which they occur within a cell. So first one, DNA unzips in the nucleus. Yes, that is the first one. Then comes mRNA nucleotides transcribe the complementary DNA message. Yes. Then comes mRNA leaves nucleus and goes to ribosome. Yes. 3. Then mRNA attaches to ribosome and the first codon is read, that is 4. So what and all over? A, D, E, C. Okay. tRNA brings in proper amino acid from cytoplasm. Yes, that is the 5th step. And a second tRNA brings in new amino acid. That is the second step, last step. So the order is A, D, E, C, B, F. That is option 3. A, D, E, C, B, F is option 3. The next one. Arrange the following landmark achievements in chronological order. Crystal structure of DNA. Okay, sequence of insulin. That was the first one. Then comes the crystal structure of DNA. 2. Then comes crystal structure of myoglobin and draft of the human genome sequence. That is the 4th one. So CADB. 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 That is option 1. Then next one. Which of the following statement about the salvage pathway for the synthesis of purine nucleotides is correct? So increased PRP levels in the cell lead to the activation of hypoxanthine guanine phosphoribosyl transferase, HGPRT. Answer is option 3. The absorption of glucose in the digestive tract, it occurs in the small intestine. Option 1. Which one is the heaviest particulate component of the cell? So it is the nucleus. Then comes mitochondria, Golgi apparatus and others. Then 93. Okay, transketolase activity is affected in thymine deficiency. Answer is option 4. Then chymotrypsin is specific for peptide bonds containing uncharged amino acid residues. Then all of the following statements about phenylketonuria are correct except phenylalanine cannot be converted into tyrosine. Now before coming to that, I will tell you PKU, in PKU, that is phenylketonuria, there is elevated level of phenylalanine and defective process in the conversion of phenylalanine to tyrosine, and a defective enzyme that is involved is phenylalanine hydroxylase, and symptoms are neonatal vomiting, mental retardation. So phenylalanine cannot be converted into tyrosine, that is correct. urinary excretion of phenyl pyruvate and phenyl lactate is increased? Yes. It can be controlled by giving low phenylalanine diet. Yes, that is also correct. It leads to decreased synthesis of thyroid hormone catecholamine melanin. That is not correct. So that is the answer. Then next one. The most of the ultraviolet absorption of proteins above 240 nanometer is due to their content of tryptophan. Answer is option 1. One international unit of vitamin D is defined as the biological activity of 0.025 microgram of cholecalciferol. Answer is option 1. The vitamin having the highest daily requirement among the following is ascorbic acid. Answer is option 4. Isoenzymes are chemically, immunologically, electrophoretically different forms of an enzyme. Yes. Isoenzymes, they are different proteins that catalyze the same reaction. They differ in their kinetic and regulatory properties, and in the cofactors they use, or in their subcellular distribution. Isoenzymes have similar but not identical amino acid sequences, and they differ in multimeric quaternary structure. So different forms of an enzyme similar in all properties? No, they are not similar in all properties. Catalyzing different reaction, catalyzing the same reaction, that is correct. So 3 is not correct. They have told it as different reaction. Having the same quaternary structure like the enzyme, they differ in quaternary structure. Then next one, a deficiency of folate leads to megablastic anemia. Answer is option 2. So that's it. Thank you for watching my channel. Please do like, share and subscribe. Thank you.